I'm Daniel Levine, and this is the Bio Report. The emergence of CRISPR, a powerful tool for gene editing, promises to correct devastating genetic diseases, but it also raises concerns about how it will be used and who will make those decisions. Neal Baer, in his new book, The Promise and Peril of CRISPR, brings together a series of essays by bioethicists, geneticists, and others who explore ethical and policy considerations raised by the technology. We spoke to Baer, co-director of the master's degree program in media medicine and health at Harvard Medical School, about whether there are unique questions raised by CRISPR as a dual-use technology. The book's recasting of the case of Chinese scientist He Jiankui, and why he thinks there needs to be public debate now over the use of the technology to make heritable changes to the genome. Neal, thanks for joining us.
It's my pleasure. Thank you for having me.
We're going to talk about genome editing, the need for public discussion about its uses, and your new book, The Promise and Peril of CRISPR. You served as editor, and this is a collection of essays from bioethicists and others that explore the genome editing technology from a number of perspectives. Genome editing is not new, although CRISPR has enabled it in a way not previously possible. Why this book now?
Mainly because we're able to do things that we haven't been able to do in the past. For instance, on the promise side, cure sickle cell disease, cure beta thalassemia. On the peril side, do gene editing of germ cells, which are reproductive cells. So we have the ability to, in a sense, change human evolution.
One of the issues discussed early in the book is concerns about CRISPR as a dual-use technology, the fact that while enormous good can be done with the technology to address deadly diseases, it can also be a tool for nefarious purposes. I understand the issue being raised, but it strikes me that you can engineer and replicate a deadly virus easily with older tools of biotechnology. Is there something fundamentally different about CRISPR and potential abuse of the technology that requires new safeguards or public debates with regards to it as a dual-use technology?
I think so. Dual use means, of course, that there are very positive things like, as I said, curing certain genetic diseases, and the problematic things, not only bioweapons, but also questions of enhancement and who will have access to these tools as they're shaped and developed further. As you said, it is true that we can make poliovirus and various other viruses, and that's not necessarily with CRISPR. That's the same, and we'll get into this, I'm sure, using CRISPR for making decisions about which embryos to use. We have PGE, where we can analyze an embryo and just not implant it if it has Marfan's or cystic fibrosis, or one of thousands of other genetic diseases. We don't need CRISPR to do IVF. What we need CRISPR for is to treat people who have genetic diseases. And the dual use side, as you asked, the negative part is that it could be easier, for instance, to make a virus more transmissible by editing genes in that virus, for instance. Right now we've seen what happens when COVID, for instance, mutates. There's a lot of concern right now about H5N1 and it's mutating bird flu. So with CRISPR, it's conceivable that one could use these newer techniques to go in and make edits that could make the virus, for instance, in the case of bird flu, more transmissible. So we don't want rogue scientists doing that, certainly. So that would be something negative. And we certainly need to talk about enhancement. We're not doing enhancement yet. And it's not about picking human beings' eye color. Genetics are very complex, and most things, most attributes are a combination of genes. It so happens that some very, very bad genetic diseases may just be a mutation of one or two letters, A, T, G, C. So that's where CRISPR comes in, where it can home in to the mutated gene, cut it with the enzyme, and then replace it with the gene that is not mutated. So I think, we need to think about dual-use technology, not only about viruses and bacteria and germ warfare, but also in terms of, as we know and learn more about genomes, not only human genomes, but animal genomes and plant genomes, that we need to be pretty much clearer about what we're doing and what the ecological effects might be. I was just reading, for instance, about a rare marsupial I'd never heard of called a quoll, Quoll, and it is in Australia. And it's being decimated by some toads that were not endemic to the area. And so these quolls are going to possibly become extinct unless, we could use CRISPR to make them resistant to that poison. Evolution might have made them resistant over thousands, hundreds of thousands, millions of years, as is the case for many different animals we can look at and see that they coexist because they've developed through the pressure of evolution to be resistant. Should we be doing things like that? Should we be taking animals that are near extinction and making them hardier or resistant to these, toad venoms, without really understanding the full nature of what we might be doing?
There's actually an example in the book around birds that were engineered to be resistant to bird flu. And what happened was they became infected by more virulent streams, potentially creating a greater risk to human populations. So
Right.
There is this issue of unintended consequences.
Yes, that's a very important point that you raised. That's in a piece about, using CRISPR to address issues in animals. For instance, should we make animals even meatier? Should we make them without horns so that we don't have to take their horns off? Should we make chickens, really with very little brain and just mostly meat? But what does that mean for the animal itself and its life? So these are raised by Marcus Schultz-Bergen, who has written a really interesting piece on that. It's not just about how we see ourselves and what it means to be human, but it also raises questions about what does it mean to be an animal, or what about transhuman issues? So, we're making organs in pigs and translating, and we're putting them into human beings now. We're transplanting them. And so does that mean that we should be doing that for human beings? Does it mean that we should be using CRISPR and putting human genes into animals? Should we be making animals more sentient in whatever sentience means in terms of, humanness? These are questions that we just have to raise before we just jump in and start doing these things.
Much of the focus of the book involves heritable editing of the human genome, editing of the genome in a way that gets passed on to future generations opposed to somatic cell genome editing. This is a practice that's widely prohibited today. The book in many ways argues for why there nevertheless needs to be public debate on germline editing. Can you remind listeners about the case of He Jiankui?
Sure. So it's a really complicated story about what happened to the doctor in China who edited the germline of an embryo that then split into twins. And what he did is he mutated the receptor for HIV. And how do we know to do that? Well, we know that there were some people exposed to HIV who never came down with AIDS and HIV. And so those were folks who had this mutation. So he went and did this, and the twins were born. And these are the first, as far as we know, human beings born who were gene edited. And that's very different from what we're talking about in terms of, say, sickle cell disease, where there have been people who have, quote, been edited. Their somatic cells have been edited. So in other words, they're cured, their fetal hemoglobin gene was turned on. That's how they did it. And they cannot pass that gene on to their progeny. So in other words, we're only addressing with somatic editing diseases that are very harmful, and that mother who was cured could still pass the sickle cell gene, would still pass if she had a child, that gene on. With germ cell editing, we can change the genes in the embryo so that they would pass on new genes. And this is what really, as you said, the focus of the book is about. And so Dr. He is the first person who did this, and it raised an uproar around the world because there's a moratorium on this. But I think we have to start looking at the history of this and understand Dr. He's history, along with some changes that are going on in terms of organizations like the American Academies of Science, Engineering, and Medicine, to understand where we've been in terms of thinking about germline editing and where we're going. So let me first start with the history of Dr. He. This is a really insightful and important piece written by Ben Hurlbut, who is a bioethicist at Arizona State University and renowned for many, many pieces he's written on science and ethics. And essentially what Ben argues is that around 2015, everyone was in these organizations was arguing that we needed societal consent, whatever that means, and we can talk about that. And we should not do germline editing. And then Dr. He did it. Why did Dr. He do it? Well, in Ben's article, he contends that he attended meetings at an unnamed university in the United States where Dr. He was. Dr. He did fellowships and postdocs at Rice and at Stanford. So he was known around the country amongst people who do this kind of work. And he was told that, like the person who invented IVF, he did it in a sort of idiosyncratic way. It wasn't through, the typical academic peer-reviewed approach. He announced it to the newspaper, and when he did it, and he was successful, and lo and behold, he won the Nobel Prize. And wouldn't that be great for Dr. He in China if he did something similar? Ben quotes an American scientist who was participating in this discussion, with Dr. He, that if he went and did this, please make him number 5 in his new company, because he certainly would start a company after this, but not number 1 or 2, because he wants the controversy to die down first before he would join a company that was going to do this kind of work. So, in other words, Ben presents evidence that Dr. He was encouraged to do some things that these American scientists knew that they couldn't do, shouldn't do, and were prevented from doing. And interestingly, after Dr. He did this, and it was announced at a big conference, I believe it was in Hong Kong, the tenor changed amongst these organizations that have geneticists and scientists. And instead of saying absolutely no editing using germline cells, maybe there are times when we could do it. For instance, maybe there are parents who both have Huntington's disease. And so then what happens is you start to make these fantastic situations up. Like, if one parent has Huntington's, they have a 50-50 chance of giving it to their child. And it's a dominant disease, dominant genetic disease. So if both parents had it, that's not a good thing. So that would be an example. Or an example is used, and this is more possible and probable, is that there are families where both parents are deaf and they have genetic deafness, and there is no possible way genetically that they can have a hearing child. And shouldn't those parents be allowed to choose to have a hearing child? These people argue, if that's what they want, and we have the means to do it. Now, the only means we have to do it is through CRISPR right now. So, because, we can't use pre-implantation genetic approaches to determination to do it, because every embryo is going to have deafness. So, we all, as I said earlier, we often argue that we can use PGD to determine whether or not an embryo is carrying a genetic disease, and therefore we don't need to use CRISPR. In this case, it doesn't matter. We'd have to use CRISPR to make a child not deaf. Of course, this raises huge questions, and Carol Patton, one of our writers who is deaf, who's a dean at the University of California at San Diego, argues that, first of all, deafness is not pathological, according to her, and she is deaf. It is human variation. And that leads us down a road of starting to discuss, and we can, what should be, what eggs should be CRISPR'd and what shouldn't, and what is pathological and what is human variation. And so this is where things are much more complicated than they certainly get addressed in the local press. And so with Dr. He, he was pushed in a way, according to Ben, to do this work because he was going to receive accolades and major support from around the world. That didn't happen, as we know. But also, and maybe even more importantly, as Ben argues, it changes the tenor of the argument from one of it's forbidden to do it to maybe there are times to do it. And, that's where I think we have some problems, because once you go down that hill, that slippery slope, it's hard to stop. And so the main thesis of Ben's work in this book is that science pushes ethics rather than ethics pushing science. And that is, I think, a wonderful example of where at first science was saying no, and now they're saying, maybe there are times to do this. And we know that in Russia, there is a geneticist who wants to do this. He wants to take deaf couples, and they're not unusual, and give them hearing children. I'm not saying that all deaf couples want to have children who are hearing, and that is, another big, question, like who should be making these decisions? Should parents be making these decisions? So that's, the background of Dr. He. He, as I said, according to Ben, he was pushed in ways that were not fair to him, and he suffered the consequences. He was in prison, and it started to open up the field in a way to consider doing germline editing. I just want to add that George Church at Harvard, and I'm at Harvard as well, argues that we should be doing germline editing because it will be, and this is in the book, less expensive in the long run. It costs, and we'll get into this, between $2 and $3 million to treat someone for sickle cell disease. We have 100,000 Black Americans in the United States with sickle cell disease, 20 million around the world. Who's going to pay for that? For this devastating disease. So George Church says, why not just get rid of these diseases? Why not make them something of the past by editing them out in germ cells?
I want to talk a little bit more about Holbert's essay because I did find it the most compelling part of the book. And in some ways, it could have framed the discussion rather than coming at the end. But he did interview her. And one of the things the essay shatters is this notion that He was a rogue scientist, as he's often portrayed. And instead, he suggests the case reflects a deep cultural problem with the enterprise of science. I'm wondering if you can talk about that a bit.
Exactly. So it's interesting that you say that, because I purposely put it at the end as kind of like, and here's where we're going, and here's what to think about. It's, this gets back to the issue of hubris and fame and success and drive, that there were scientists at these meetings with her who were pushing him in ways to do what he did because they knew they couldn't do it here. But maybe they thought that, and not maybe, I mean, it seems to me that that's the reason they did it for this reason, because they knew if it got done, it would make it easier to do it here. And so that's really, the issue is that once something is done, it's hard to undo it. It's hard to unsee it. And that's what we've seen with this now. And we're seeing this. Renowned scientists, deans of medical schools are all saying, not all of them, but many of them are saying, maybe we should consider times when germ cell editing is appropriate. That they were not saying this in 2015. They're saying it after Dr. Hood did it. And then they're finding rare examples to support and undergird their approach. That gets us to some issues about oversight. So Andrew Heinrich, who's a lawyer, talks about oversight. Have you ever heard a politician? Have you heard those running for president in the United States? Harris, Trump. Have you heard senators talk about CRISPR? No. You hear them talking about AI all the time, and fears about AI, but nobody talks about CRISPR. Are there controls over CRISPR? Not really, in terms of the buying the DNA pieces. We look to the companies that manufacture these segments to oversee to whom they're selling them. There are certain segments, because we know what's in polio and smallpox, and we know those sequences. So we know if somebody's buying them for a lab, that we need to, monitor them, and we turn it over to those companies to do it. But there are no laws that force companies to not sell certain things. There's no international law. There's discussion, but it's not discussed because it's complicated, and people just think of gene editing as something that's sci-fi, and we're talking about the ability to do just a few things right now, but those things that we can do are pretty fantastic. The fact that we can cure a disease that is so devastating to children, like sickle cell disease, which really shortens lifespan, or beta thalassemia. And we're on the verge maybe of curing progeria. There was just a piece on that. That strikes very few children, but still is a devastating disease, that speeds up their age. They die by 20 often. The sky's in a way the limit. Will we be able to cure CF, cystic fibrosis, or Duchenne's, these really tragic diseases that, often strike children? Everybody would want to do that. A lot of us think maybe we should use PGD to just not implant these, but that's also controversial. And so, and then we get into this issue of what's devastating and what's not. So, for instance, in Denmark, any pregnant person can have a test for Down syndrome, and they've not eliminated Down, but they have very few children born with Down syndrome. And so we have an essay, a number of essays, but one by Rosemarie Garland Thompson, who's renowned in this area of disability rights and ethics, writes about what she calls velvet eugenics, and talking about kind of starting to, get rid of people from being born with Down syndrome or other genetic syndromes that one can live with. And, we can't predict when someone's born with Down syndrome whether they'll have a full life. I'm a television writer and producer, and I did a show on Law and Order Special Victims Unit with a woman who had Down syndrome, an actor, and she was in her mid-30s, and she passed away sadly a few years ago, early 50s, from Alzheimer's, which strikes down people often when they're younger. But she lived a full life. She was a performer. She was on television shows, and she was able to live on her own. Not all people with Down syndrome can do that. There's a range. And so we can't predict who's going to be, living a fuller life and who won't. And so we're seeing fewer people with Down syndrome being born. Of course, this gets into political issues in the United States, of course, on abortion and as well, and selective termination. But the point I'm making is that where do we draw the line on what's a livable life and what's not? Certainly, if you're a child and you have adrenal leukodystrophy or Tay-Sachs, that is not a livable life. That is a life of suffering and a sure early death. But other, cystic fibrosis, we have a piece by Sandra Soufian. She's in her early 50s, and she argues in a really profound way about what she's gotten from having cystic fibrosis and the community that's developed. She's not arguing that it's a good thing to have CF, but she's asking us to think more deeply about, what we're all saying as physicians, like I am, that we want to cure people, but, and she's asking us to think about the lives people lead who have these genetic syndromes. So, that's why I find this all so fascinating, because it raises so many fundamental questions about what does it mean to be human? What does it mean to be a social person? What does it mean to have a life that's, lived with a disability? We'll all be disabled at some point in our life, unless we die of a stroke or of a heart attack, right off. And so it's just a longer period of time for many of us than for others. So I think CRISPR brings up these fundamental questions about what does it mean to be human? What does it mean to have the ability at some point down the line to control evolution? Now, we can't control evolution. Now, we can't, make people stronger or smarter, whatever smarter means. Those are complex interactions between the environment and genetics and many genes. But we need to think about now what we're doing because it will have a profound impact on the future. And we have a number of pieces on this. And I think this is really also a key topic in our book. Plastics were going to save the world. And of course, they've made our lives much easier. But now we're starting to see the consequences, not only in the ocean and killing sea mammals, fish, but we're seeing it in microplastics. And what does that mean now for human lives? So many things this is the dual use technology. Again, many things that have been developed as a promise for the future, gasoline, cars, have serious consequences as well. And we weren't able to predict them so clearly, though there is evidence that the oil companies did know a lot about climate change for many years before they really admitted that they did in burning fossil fuels. So this book is asking us to think now and examine what we're doing before, before we know what the outcomes are. How do you do that? That is really tough. Scientists like to do their work. They don't like to have folks overseeing them, putting restrictions on what they can and can't do. And so how do we think about this as a society? How do we think about the future? Michael Sandel wrote a piece many years ago in the early 2000s for The Atlantic on gene editing, and he was very prescient. And he talks about the enhanced and the normal, and what does it mean to be human, and what does it mean when we're able to maybe make someone stronger. We do know, for instance, that there are certain genetic anomalies, like there are children who don't feel pain because they have a mutation. And one I read recently, a child died in India. He was performing for friends, jumping off a 4-story building, and he had broken his bones in the past, never hurt. Well, he died doing this, but we know there is talk, I don't know if it's true, that Russia and China are doing research on soldiers to make them pain-free, sleep less, maybe less fearful. How do we look at the genes that are involved in those parts of our being? Do we want to be manipulating them? So these are the questions that that CRISPR raises. And yes, we've had zinc fingers and things like that we could use in the past to do editing, but it's just becoming easier, and we're always worried about what we call off-target effects, but we're better at that. Scientists are much better at preventing unpredictable consequences in terms of affecting other genes in the genome when you CRISPR a certain gene.
I want to go back to Holbert for a moment. One of the things he does is challenge what I'd argue is a widely held belief around these discussions that science advances faster than our ability to regulate it or address the legal and ethical concerns. Do you think he's correct?
Absolutely, because he's raising it now, and we're having this conversation about, should we have oversight of CRISPR? Should we have laws that make companies keep very strict track of what they're sending out to labs and to scientists? scientists are pretty free to do, what they want to do. And I've talked to some who say, we can monitor our own. You know, I've had an argument with one about this, that they'll be seen as a pariah if they do something wrong. Well, that's all well and good, but that doing something wrong could have dire consequences. So I think Ben is absolutely right that science tries to take the lead and then tell us what we should think. Now, and I think that's been really well illustrated with this Dr. He example, that we have changed our views. And I say we, many scientists have changed their views, geneticists, physicians, about germ cell editing after Dr. He did it. It's like, well, it can be done, so, let's go forward. That's not to say that I don't believe in what science does. I mean, I'm absolutely, I'm a pediatrician. I am pro-vaccine. I am, very much interested in improving the health. I have a program that I created at Harvard Medical School. It's a Master of Science in Media, Medicine, and Health, and we look at how to tell stories to improve health. And that's why I am all in favor of somatic editing. Now, again, we haven't really delved into this. Who's going to pay for it? Vertex has made an amazing treatment for sickle cell disease, Bluebird for sickle cell and for beta thalassemia. How are we going to pay for, those who qualify, who can have this treatment? 100,000 people in the United States who suffer. These children have strokes when they're 9 years old. They're in constant pain. They're treated as pain-seeking often because the kinds of treatments that we have really don't, assuage the pain in ways that we would want it to. So how are we going to pay for this? Is our insurance companies going to pay $2 to $3 million per person? What about the 20 million people who have sickle cell around the world? And here's also what's interesting about all this too. It's like, why is there sickle cell? Why is there this terrible disease? Well, we think that, first let me say, 1 in 13 Black Americans has the trait. So when you have the trait, does it really affect your life? No, because you have you need from each parent the trait. So 1 in 4 chance of having a child. If both parents have sickle cell trait, 1 in 4 of their children, on average, will have the disease. So how are we going to, so why did this occur? Well, we think it occurred because in Africa, having the trait seems to be protective against malaria. So here's where evolution is involved. So those who had it would survive more likely if they had the trait, but sadly, those who get the trait double die. Evolution doesn't really care about that so much as just survival. And so we see this. Interestingly, I've read with Dr. He that people who have this mutated receptor for HIV are more susceptible to other viruses, like I believe West Nile virus. So evolution had in a sense, a plan, not like a plan, like what we're going to do on our schedule today, but the pressure, of course, of the environment on human beings or on any animals shapes us. And so with, when we start to, take this quoll, this marsupial or a human being, and start to address and mutate and address a mutation, what are we doing? Do we understand fully the ramifications of why this mutation exists? it was a great mutation to have if you were exposed to HIV, but might not be such a great mutation to have if you're exposed to other things. So knowledge is critical, and we're just not there yet. I just want to add another interesting point about this, which is Kevin Esveld at MIT invented the gene drive. And a gene drive can be used to make, it can be introduced into like mosquitoes, for instance, and make them infertile. And it works in populations that have a fast reproductive turnover rate. So conceivably, we could get rid of Anopheles mosquitoes that cause malaria. But I think we've hesitated, though there are experiments, I understand, in Key West to doing this, because we don't really know the whole ecological consequence. There was a really interesting piece in the New Yorker some years ago about this, and about Kevin going to, I think, was Martha's Vineyard and or Nantucket, where there was a Lyme disease, and a discussion amongst the citizens there about whether to introduce a gene drive into mice to stop the Lyme disease from reproducing through mice, deer mice. And I think the population or the folks there voted against doing it. And it was interesting approach because it's like a kind of it's an island, so it's not lesser chance of having spillover, but still I think people were concerned about the ecological ramifications. Like, what does that mean to stop this? Diseases are bad, but diseases don't have feelings. We have feelings. And so diseases just want to survive. And we've seen that with COVID. And I think we're going to see it with H5N1, and we better be ready. So, so that's, again, another element of this CRISPR conversation that I think is important, and we need to be talking about it much more than we are.
Well, where do you think we are in terms of the acceptance of heritable genome editing or the lifting of prohibitions on it?
Well, I think there's not a lot of, as far as I've read and seen and talked to experts and the writers who worked with me, against, none of the writers that I worked with are against somatic gene cell editing. But they do raise these really tough questions. For instance, one of our writers, Dr. Weiss from UC San Francisco, is a cardiologist, Ethan Weiss, and his daughter has albinism, genetic disease. And had he and his wife known when she was pregnant, he writes in his piece that they likely would have aborted the fetus. But they didn't know, and their child was born, and she has many health challenges, but she's changed their life in ways they can't imagine positively. And so it is this complicated situation of, what we know now, having our daughter, makes us say that, we have to be very careful about what we're doing, in the future. But, when we didn't know, we probably would have done something where we wouldn't have our daughter. So it just raises these fundamental questions about, what can we know? What should we be doing? And should we take, a step back and just start talking about it before we keep going forward and saying, well, maybe there is a reason to do germ cell editing now because this couple who is deaf wants a child who isn't deaf. And then people might say, well, who are you to say what this couple should want? If this is available, why shouldn't they get it? And so, and then we have people who are deaf saying, I personally don't see my deafness as a disability. I see it as, who I am. And so I think one thing that's important about CRISPR is it broadens our view of inclusiveness. And, instead of just going out there and saying, well, we're going to have to cure all these people, maybe we have to take a step back and say, one, do they want to be cured? And two, should we be, getting rid of genetic syndromes? And again, where it gets complicated is no one's going to disagree. I can't imagine that we shouldn't get rid of Tay-Sachs or adrenoleukodystrophy, or Huntington's disease, two horrible diseases of childhood, one of middle age, that cause horrible suffering and death. And so, where do we draw that line? What does it mean to have a full life? These are fundamental questions that I think we don't talk about now. We talk about culture wars, but we don't talk about what it means to be human. What does evolution really mean? Should we be in control more of our destiny, of our bodily destiny?
As we mentioned, the book focuses largely on heritable genome editing and the issues around that. You touched on the issue of cost and access when you were talking about sickle cell disease, which, is a great example. But even with somatic cell genome editing, there are questions about access and cost that you don't really get a chance to talk about in this book. And when you put it in the context of global health and a disease like sickle cell, for which we've already seen a CRISPR therapy come to market, how do you think we should address issues of access and affordability, even though it's not a problem that's limited by CRISPR, but again, it
Right.
becomes ever more acute?
Well, it's a really tough question in terms of, looking at the business side. But I think that healthcare is a human right. And access to healthcare is a human right. And so this exists. So what is the cost? Why is it 2 to 3? We have to start asking these questions. Why does it cost between $2 and $3 million? Is that the research cost that it cost Vertex to make it? Vertex wants to make a profit. Should the government be paying for these treatments and make sure that Vertex recoups its investment? But, it's a capitalist society. And our healthcare is profit driven. And so, even though I support healthcare as a human right and access and equity, I see the barriers. And I do think every person who could qualify, there are certain people who can't get it because they've had occlusions or strokes and things like that. But people who can get it, I think they should. And there's discussions about how can Medicaid pay for this and address this. But this is the tip of the iceberg, because we're also able to cure beta thalassemia and some very, very rare protein folding diseases of the liver. It's easier to deliver CRISPR through the blood, so that's why it's easier to treat these diseases versus CF in the lungs. And CF is complicated; it's not in every lung cell. So, how do we address a profit-driven system of healthcare where pharmaceuticals cost a fortune? 1 out of every 2 Black men who have sex with someone who's HIV positive, sex with men or trans men or others, 1 out of 2 in the United States, in the South, is HIV positive. How is this possible, given that we have we have treatments to prevent, we have PrEP to prevent HIV, and we have treatment to make people undetectable. And if you're undetectable, you're untransmissible. So how is it that we have almost 1 out of 2 in Los Angeles, 40% of Black men who have sex with men are positive? How is this possible? Stigma, access, cost, shame, the church. But still, we have fairly inexpensive compared to treating sickle cell, ways of preventing it, and yet we failed. And so, how are we going to keep introducing CRISPR-based treatments, personal-based medicine, when we can't afford to treat, how are we going to treat 100,000 people or 20 million people around the world with sickle cell disease? That's a question of the whole system of how we provide healthcare, both in the United States and around the world. So these are big questions, but are we just going to say, well, if you have insurance and they'll pay for it, great. Or if you're wealthy, great. Is that how we should go about this? Again, these are conversations that we don't have, which we should have, because, I think the healthcare system has obviously failed LGBTQA plus Black men in the South, for instance.
Well, what would you like to see as a way forward in terms of a public discussion on CRISPR? And do you see any organizations or forums where this type of public discussion is taking place?
Well, I think there can be many different forums, and I know that there are certain organizations that do this. So, for instance, Marcy Darnofsky, who wrote a piece called Twisted Tales in the book, is the executive director for the Center for Genetics and Society. So they do programs and publications and discussions and bring folks together. We need to have, of course, the national academies talking about this, not just from the side of, what CRISPR can do to treat people, but also the ethics behind it. And I mentioned briefly in my piece, drawing on the arts and humanities, which is what I do at Harvard Medical School, where we look at any art approach you can think of, be it dance or music or poetry or documentaries, and we look at how do we tell the story, always drawing on the data first, to provide better healthcare for people. So I call it going from the data to the emotion to the action. And so, we look at the data, say, as I did, 1 out of 2 Black men who have sex with men being positive in the southern United States. The emotional approach would be, there was a wonderful piece by Linda Villarosa a few years ago in the New York Times about this. That's how I learned about it. And then how do we take action? Do we do it through policy? Do we do it through community organizations? So we draw on, the art there was the essay, but we look at all different ways of telling the story. So for me, what I plan in terms of CRISPR is to do some vignettes based on many of the pieces in my book, because I'm a writer, a television writer I did ER and Law and Order Special Victims Unit, and Designated Survivor recently as the writer, producer, showrunner, that I develop stories. For instance, I did it on Designated Survivor. I think we did the first CRISPR-based storyline. So we tell stories, and stories appeal to us. Like, why do we love novels and movies and music? Because it emotionally affects us. And so this is a way to, I think, have conversations. It's not just like we're going to have panels, but I think we should draw on the arts and humanities to tell stories that I've been telling today. For instance, Carol Patton is deaf, and she could tell the story about someone who's deaf who doesn't see themselves as having a pathology that needs to be fixed. That's something that isn't seen a lot in movies or television or in other artistic or humanities forums. And it would be great to see that. Now, there are people who are deaf who want to hear, and they have a right to do that as well. We have cochlear implants, but it's not just one way or the other. And I think that's where the arts can come in and help us to understand, the complex issues at hand here. And it is gray. I'm not saying that I'm for or against using CRISPR to treat an embryo. I mean, I'm worried about treating embryos from deaf parents to make them hearing because I worry about what comes next, a la Ben's piece. But I certainly understand, I have a son, I certainly understand if someone wants their child to be hearing. And is it my right to say no? But at least let us talk about it, as opposed to having scientists and geneticists doing it and nobody knows what they're doing.
The book is The Promise and Peril of CRISPR. Neal Baer, editor. He's also the co-director of the master's degree program in media medicine and health at Harvard Medical School. Neal, thanks so much for your time today.
Thank you for having me.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.